Hi everyone, my name is Brad Balderson. I'm a second year PhD student in Mikkel's Burden Lab at the University of Queensland. So this is a presentation that I made for the course Sci 2100. Um, so it's called Approaching Bioinformatics, or How to Get Good Grades in the Course. So the real inspiration for this is from Sydney Brenner, who's a Nobel Prize laureate in cellular biology. So he said that a proper simulation would allow us to make predictions by performing experiments on the model and calculating what it might do. Not only must we use the vocabulary of the machine language, but we must also pay heed to what may be called the grammar of the biological system. So Brenner said this in a perspective article he wrote called Theoretical Biology in the Third Millennium, which was published 3 days before the new millennium, so 3 days before the New Year's. So it's very good timing. So the key aspect, I think, when approaching bioinformatics is we need to think of it as representing biology computationally, which is what Brenner was getting at when he meant using the grammar of the biological system. So thinking about this just from the central dogma, we have this model of going from DNA to RNA to protein, and transitioning between those is facilitated by transcription and translation. But how we actually represent this in the course is by using object-oriented programming. So we have our sequence objects, which stores, for instance, DNA, RNA, or protein sequences. And whether or not it's DNA, RNA, or protein, it'll have an alphabet which specifies which one of those, and it stores a string of molecules. And then if it's a DNA, then we can transcribe it, and if it's an RNA, then we can translate it into protein. So that's just an example of how we go from this mental representation of the central dogma, and then we subsequently express that mental representation in our code. So this is also evident later in the course when we start thinking about evolution. So this is a famous sketch from Charles Darwin when he was figuring out his theory of evolution, and he's actually got a phylogenetic tree sketched out here. So we represent this in the course using the phyllo tree. So we actually have an alignment object which represents an alignment of sequence objects, which I just mentioned earlier. And then you can calculate the distance between these sequence objects to create a phylogenetic tree. And when I say distance here, I mean some kind of measure of evolutionary distance between those sequences. And then on that tree, you can perform certain analysis, such as ancestral sequence reconstruction, which is what Brenner was getting at 20 years ago now, when he was mentioning about performing experiments on the model. So now I'm going to go through a general approach to solving bioinformatics and I think most other problems. And as a result, this should help students do well on practicals and exams. It's good to have a good guiding philosophy. And this has helped me quite a lot with my postgraduate work. So I think that the best place to start is with a mental model on how you actually think the biology works. So you can get this from lectures, from reading, from writing, from drawing, from observing, from dreaming, from thinking. It really helps you develop the mental model of how the biology is actually functioning. So you create a kind of mental simulation. So this is evident when I mentioned earlier about Watson and Crick, when they're looking at some of the X-ray crystallography data, that they ended up coming up with this kind of mental representation of how they think biology was actually working. And then you've got Charles Darwin, who had all of his observations from his trip on the Beagle, looking at, for instance, the finches at the Galapagos Islands. So once we have this kind of mental model, it takes a bioinformatician, someone with some programming skills, so you can actually represent that mental model computationally, either through object-oriented programming or some other way of representing the biology within your computer, so you can perform those calculations on the model. So Mikkel wrote the Sci 2100 course, and he came up with sequence objects, phyllo trees, ancestral sequence reconstruction, etc. And that's these are the objects that the students use and manipulate throughout the course to get a feel for how how to actually play with computational representations of biology. So once you've actually got these computational representations, you make new testable predictions. So that is, you're performing simulations based on the model. So Gabe Foley is a tutor in the course and almost finished his PhD student, PhD in Mikhail's lab. So part of Gabe's PhD work is ancestral sequence reconstruction using this tool called GRASP. So they actually, for this particular protein family, actually predicted what the ancestors were, and then predicted what the ancestors were for these. So you could see in an evolutionary sense what changes were happening in this particularly important enzyme family to try to get a sense for what makes the different present day enzymes different from one another. And then once we've got this new these new predictions, you actually want to test those predictions, and that's where it's taken to the wet lab by our biologist friends such as Connie Ross, who's in Elizabeth Gillam's lab at the School of Chemistry and Molecular Biology at the University of Queensland. So she actually took those ancestors and recreated them in the lab. She measured their thermostability and also enzymatic activity, and actually found that those predicted ancestors were actually functional. They had increased thermostability, and they also had enzymatic activity, which is pretty remarkable considering the sequence space. So what aspects of this sort of process do we test for in Introduction to Bioinformatics, Sci 2100? So thinking about this in terms of having a mental model, we express that mental model with code, we make new testable predictions, and then we test those predictions. So in our Sci 2100 course, we're testing the mental modeling, so with your lectures, etc., and then expressing with code. I would give half a smiley face to this because we're mostly just trying to understand the code. And then we make new testable predictions. So that's only about 1 out of 10 smiley faces, and then 0. We leave that to the biology courses and to our experimentally skillful friends. So now it kind of gets to the part of like, how do we actually use this to get good grades in the course? So there's this really awesome saying by Charles Goodhart, who's an economics Nobel Prize laureate. He said that when a metric becomes a target, it ceases to be a good measure. So what he actually means by this is, if you focus on grades and actually forget the importance for what those grades are trying to measure, then it actually can make things quite make it quite anxiety producing, because you start saying things like, what's going to be on the exam, what are they going to test me for, what's expected of me. But what we're just trying to measure is just your understanding of bioinformatics. So really the best way to get good grades is to just forget about them. They're just measuring your level of understanding. So the best thing to do is just forget about those things. Those are just good feedback for how well you manage to understand the content, and instead just focus on this mental modeling aspect by about 1 smiley face, and then this practical skill level and understanding how to make new predictions. And if you just focus on trying to understand it, everything becomes so much more enjoyable, because you're starting to see how really impactful this sort of approach is in actually revolutionizing biology. So it makes it far more exciting. And then when you just take the exam, it doesn't matter what we ask you, because you understand it like the back of your hand. So you're going to do well regardless of what happens. And I really want to make this point here about really focusing on the joy of learning this sort of topic and continuing your bioinformatics journey is, could we simulate the universe? Because what we're doing here is in this case, we're just taking biology mental representations and then expressing those within our computer in code. But technically, you could do that with just about anything, right? You could represent, people do that with the molecular dynamics simulations, where you actually simulate every atom in molecules and then let those molecules interact to understand the biophysics of the system. So it might be even possible to simulate the universe in the future when there's enough computational capacity. So it really makes me wonder, what do you have to offer here? What's the mental representation that you have of biology that then you could be representing into your computer and to test how the world works better. And I think that is seriously amazing. And I think when historians look back 50 years from now, they're going to be thinking this bioinformatics thing was a big deal. And a lot of the other computational disciplines are a big deal as well. So with that in mind, I'd like you to think about this, like to think about what you could be helping us understand, because really in science, this is what we serve here, is this collective mental understanding of how the universe works. Just trying to perceive how things really work. So I had a few things to help understand these different parts. These are the courses that I took to help me develop some of these aspects of the bioinformatics method. So going from this mental model, these are courses at UQ. So for proteomics, this is BioInf 2000, 3000, and genomics is 1020, 1040, 2202, and 3006. I'll just leave this up here briefly, just because it's not going to be particularly interesting to people who don't go to UQ. But for software engineering, this part is extremely important. Your capacity to actually represent the code, that's where the real difficulty comes in. There's lots of biologists that are really awesome in the wet lab. I have no idea how to do wet lab stuff, but you want to be practicing your capacity to actually represent that biology computationally. I think that's the difference between a biologist and a bioinformatician is the capacity to represent the biology computationally. So a good course at UQ for this is, well, advanced bioinformatics. We mostly go into biological modeling. Hopefully that will be renamed soon to biological modeling. And CSSC 2002, which is programming in the large, which is mostly focused on object-oriented programming. And there's this really great course called Probabilistic Graphical Models by Daphne Koller at the University of Stanford. It's on Coursera. And this revolutionized how I look at the world, because it's really, I think, the mathematics of biology. It's representing processes that are inherently stochastic within your computer in a way that's computationally tractable. So these probabilistic graphical models are extremely powerful, and they mostly fall under the bracket of Bayesian networks and Markov models. There's also the machine learning courses at EQ, such as algorithms and data structures, which is really useful. So you can actually run those simulations on your computational representation of biology. And there's also artificial intelligence. So that's actually really useful when you're doing things like training the model to start with, and doing search algorithms for different possibilities. So these are really powerful to improve your capacity to represent biology and actually run predictions so that you can make new predictions about how the biology is actually working. And then a lot of third-year biology will probably be focused on testing those predictions. I don't have much experience there, so I can't really say for sure which ones are good to take. So with that in mind, I want to point to the power of the bibliography. These are some influential sources that really helped me develop my understanding of how I think bioinformatics should be approached, which is, the main one is Sidney Brenner's Theoretical Biology in the Third Millennium. Sidney Brenner had a huge impact on biology and was a huge advocate for the Human Genome Project. So, and he also established C. elegans as a model organism and was a big advocate for model organisms in general. So we owe a lot of the new developments in biology to the influence of Sidney. This book is really awesome. It's called How to Solve It, a New Aspect of the Mathematical Method. It's published in 1945 by George Polya, who is quite a renowned mathematician, I think at Princeton University. So it says mathematical method, but the examples are just given in mathematics, but the introduction to each component of the problem-solving method that George goes through is presented in a very general way. So I just skip over the mathematical examples, and I just imagine an example in a problem that I was solving recently, and I realized a bunch of mistakes that I was making in terms of the problem-solving process, and I realized a bunch of places where I was my thinking wasn't quite correct. A good source of inspiration is Einstein. Einstein was the master of mental modeling and simulation. His first paper He had no references. It was all thought experiment. And then at the end, he said, "Can anyone test this? This is a testable prediction." He did this while working at the patent office, so he couldn't even get a job in a university. He he had a lateral move. He went from person who couldn't get a job in university to be a fully fledged professor. So this mental modeling idea is a seriously powerful technique. Einstein called it thought experiments. So there's also Claude Monet's technique for impressionist art, which is, I think, very much related to Einstein's thought experiment techniques. So Monet, the impressionist artists, they focus on actually creating an expression of the world outside to try to capture the liveliness of the picture, similar to this painting that I've got in the background here. So Monet's technique for doing that, he was the master of this. He would sit at a window and he'd look outside at the scene, and he'd then shut his eyes and try to visualize the scene as vividly as he possibly could, and try to get every detail perfect. And then he'd open his eyes again and he would test whether or not the visualization he held in his mind was detailed enough. And then he just repeated that for hours on end until he could recreate that image extremely vividly in an immense amount of detail. And then he would begin to paint. He would begin to express the image that he held in his hand, in his head with painting. So it's a very similar idea with bioinformatics, except we have this image of how biology is working, and we try to express that as a computational representation so that we can then perform experiments and discover new biology. And that's exactly what Einstein did too in physics, except his form of expression was mathematics. In our case, our form of expression is computer programming. A Philosophy of Software Design by John Oosterhuis. John is a professor of computer science at, I think, Stanford. This corrected a lot of mistakes I was making in my programming software design, and this was really useful. It's only about 200 pages of it's 200 pages of pure gold for how to do proper programming. And it corrects a lot of common mistakes that you find in programming courses. The Pleasure of Finding Things Out by Richard Feynman is quite nice too. I won't go into detail about that, but also for helping with the mental representation, Molecular Biology of the Cell by Alberts and others. I read that quite often, trying to understand how the biology is working, and it's a seriously good book. You can try to read the literature to do it, but a lot of the time the literature, the scientific literature, it's all spread out in the different places, whereas Molecular Biology of the Cell is quite vivid and has nice schematic illustrations to help visualize what's happening. Probabilistic Graphical Models by Colin Friedman. So I mentioned that earlier. It's pretty much the mathematics of biology. This is going to be a seriously big deal, and I think that every computationalist should be trying to learn about these probabilistic graphical models. They're very much related to common machine learning techniques, except they're more interpretable because you're the one who actually sets up what the model looks like based on your mental representation. So they're completely interpretable. And my mentors, Mikael Burden at the University of Queensland, and Stefan, who's a bioinformatician, and Stefan Thor, who's a developmental biologist at the University of Queensland as well. So getting some good mentors is seriously helpful to develop your philosophy on how to approach science, and in this case, bioinformatics. So there's no questions because this isn't live. But hopefully this is helpful to someone, and hopefully it provides some motivation to learn bioinformatics. It's an extremely awesome field and extremely rewarding. I really love learning about biology, and I really love programming. And if any of those things appeal to you, then definitely check bioinformatics out. Thank you very much for listening.